you see bioinformatics software, as I told you in very earliest, bioinformatics what? It's a combination of biology and the computer science. Computer science is like in a sea. Okay, it's like an ocean. You cannot say that computer science means something word Excel or PPT, something like that. Come here, if you want to work on bioinformatics, uh, these are the certain software, which in software you can say these are the certain term we should know. Like suppose I'm just tell you one of the word here is a next generation sequencing analysis tool. So again, for this software, you must know, like a, have you heard about the Illumina, Tort? These are the everyday. There are some software as available or the coming in the market, where we can where we can do some work in like a RNA sequencing or DNA sequencing. In the same way, there are tools for DNA, RNA, protein sequencing analysis, including like alignment, assembly, and multi-finding and blast, and class 2 and FASTA. So what I think, there are a lot of things initially you have to know. Yeah, suppose somebody is interested to structural analysis, then he has to know what is PyMol is in a software. You have to learn at least once. And I don't think so. This is a too difficult to know, too difficult to learn. Yeah, like in a natural deals, like an organization are available who can help you to make you learn about this thing, how to run the PyMol, or how to learn the Swiss DDB viewers, or how to like how to perform this kind of work. A lot of software, as well as a lot of like a database or data bank are available. Even you don't know exactly, if you want to check particular gene, though it is available, the sequencing has been done, but you don't know how to approach, or where to go to get exactly the detail of that gene. So these like natural deals, like organization will let you know you have to go first to the NCBI, and from like DNB and such and such, their tools are available, such and such websites are available where you can get the free information regarding the gene and the chromosome. Programming, yes, of course, for doing research, dry lab, you must have you must learn something about the certain language. So you must know what is Python, what is though Perl is very old, but still people are preferred to learn, and Java, C++, and others are. It's not very it's not very essential part of the dry lab, but if you learn, it's a good one, because when you are doing some work, now you see, oh, that work has to be done by this one, that work for doing that work, I must have learned like a Java. So it's okay. There are in dry lab, there are a lot of things which you need to know, but once you come to know na, I don't think so there will be future any difficulties in the future. Well, I guess as I told you, even you don't know, if I want to go to that, I want to access certain particular genes, so where I have to go. So these basically, how far you study? You studied like if you suppose if you've done the PhD in zoology, PhD in microbiology, but these are different. These things must have informed by your instructor very first of all, like a PDP, protein database, NCBI, by BLAST, है. So these are the basically what there are certain database where you get a particular information regarding the gene, protein, and all this thing. So naturally, there is someone who will instruct you. Uh, if you want to go, if you want to get some information regarding the protein, but for that you have to go to PDB, protein database, and how to access, not only that, and how to access the protein data bank. Because suppose somebody tell you, you can open, you see Either you have to do a hit and trial, then you hope after 6 months you come to know, okay, oh, to know the simple one, this one, it has to be done in this way. But if there is if, but if there is someone who can tell you, this is a proper way by which you conduct also work. Yeah, this same. Okay, there are some, software algorithm you need. You again, you need to learn something. All this thing. Well, here, you know, this is the basic. We all know very well what are the latex and all thing, the tools for creating and formatting research paper, reports and documentation. This is, whether you do the wet lab or whether you do the dry lab. But when you convert whole data, you have to convert some. Suppose after doing a research you need to publish one paper. After doing research you need to make a draft and a manuscript for publication in a good journal like an Elsevier, Springer. So obviously you need to convert you all data in either word or in somewhere else. So for that you need to know all the basic things of how to run the latex and what and all this thing. That is very basic. Whether you do the wet lab or whether you do the dry lab, you have to know these basic things very well.